Swayam Prabha. Digital India. Educated India.
Welcome to lecture 9 on the course on bioreactors under the NPTEL online certification program. Let us, in lecture 8, we looked at the solution to the practice problem 2.2. We are in module 2, which looks at the two major outcomes from bioreactors, the biomass and the products, and some enzyme related aspects, the products from enzyme reactions. Let us move forward in that module. Most likely we would complete the module in this lecture. The last aspect that we are going to see in this lecture is the concentrations of biomass or cells, substrates and products, the measurement methods. We saw how crucial the information on biomass concentration, substrate concentration and product concentrations were in terms of kinetics, in turn the design and operation of relevant reactors. Therefore, we need to have good measurement methods to find out the concentrations of biomass cells, that is cells or biomass substrates and products. We would look at some of the common measurement methods that are used for these. There are two kinds of methods, one online and the other offline. Online means that the bioreactor is not disturbed. It is measured online as the process goes on without any disturbance. Offline, we take a sample from the bioreactor and then measure it away from the line or away from the bioreactor, and that is why it is called offline method. We will look at some online methods. They are of course preferred because they are measured at the point of interest without disturbing the bioreactor. So the best view or the best information comes from online methods. But few are available for reliable measurements, and that becomes the difficulty with online methods. For example, if we look at measuring cell concentration, there are a few online methods. There is a fluorescence method that measures the fluorescence of a molecule inside the cell. It's called NADH. You all know NADH, nicotinamine adenine dinucleotide, the hydrogenated form of it. NADH is fluorescent, NAD is not, and the level of NADH is a direct indication of the cell concentration. Higher the NADH fluorescence, higher the cell concentration. The relationship is known has been established. So you could follow the increase in cell concentration through the NADH fluorescence, or it's also called the culture fluorescence, the NADH fluorescence change. What is typically done is the fluorescence measurement probe is placed in a special well in the bioreactor. This is an optical probe. Before that, I need to tell you that NADH, the optimal wavelength pair for NADH is let's say 340, 460 nanometers. So the excitation wavelength needs to come in at 340 nanometers. The emission wavelength needs to be measured at 460 nanometers. The in a spectrophlorimeter, you have a nice cuvette or some other well kind of a measurement. You have a controlled way of sending in 340 and measuring it at 460. Whereas in a bioreactor, typically what is done is a fiber optic probe is interfaced with the spectrophlorimeter. The fiber optic bundle, some bundles carry the excitation wavelength from the spectrophlorimeter and project it in an open-ended geometry into the bioreactor. And then the emission wavelength at 460 is collected through the same fiber optic probe taken back to the spectrophlorimeter and measured. This is called an open-ended geometry of measurement. Well, those are the details. The main point here is that the cell concentration can be measured by following the culture fluorescence or the NADH fluorescence. The conductivity of cells can also be measured. There are some probes for it. The reliability, not so sure, maybe. That can also be used. That's an online method. For the substrate and product concentrations, there are a few online methods. As I mentioned earlier, the optical fiber is integrated with a spectrophotometer or a fluorimeter, or you could take a sample from the bioreactor as it is operating, but bring the sample back into the bioreactor after it flows through what is called a flow cell, which is placed in the fluorimeter for measurement. Okay, this is another way of doing that. You could use absorbance fluorescence for some substrates, some products. What is recently of interest, it is what is called near infrared spectroscopy. In fact, we have a paper on this, which was the work was done by Sanjay Tiwari. It was developed as a part of his PhD thesis and was also applied in the industry. The advantage here, let me talk of this specific case so that you will understand the advantage. Here, this industry was looking at using low levels of substrate because the substrate was toxic to the cells for their own product, commercial product. So, the substrate needs to be maintained at a certain low concentration, of the order of about millimolar and so on. I think it's about 2.5 to 5 millimolar. So, if hold on, it is substrate concentrations need to be measured, need to be kept at a low level, and also the product concentration needs to be maintained at a certain level, otherwise it would become toxic. So, let us concentrate on the product here. The older method that was followed was something like this. They would take a sample from the bioreactor. This is the production reactor. The measurements such as HPLC and so on are available in the lab, which is at a distance from the production unit. The production unit is large. Therefore, they need to take a sample from here down there, measure it. It takes time for measurement, maybe about 20-25 minutes, and then bring it back. By that time, the product concentration would have gone up and they would have lost the batch. It would have become toxic to the cells and they would have lost the batch. So, to avoid that, they definitely needed an online measurement which would immediately tell them what the concentration was, so that they could take appropriate action to maintain the product levels at acceptable levels. So, to do that, Sanjay had worked on NIR spectroscopy and developed a method for doing that, and that obviously was very beneficial to the industry. That's a paper there. It was published in Biochemical Engineering journal. If you're interested, you can take a look at that. Also, the gas that comes out of the bioreactor, we provide air for most aerobic bioreactor systems. The exit gas, which consists of CO2 and O2, and of course nitrogen and so on, that can also be measured to provide some input into what is happening in the bioreactor. So, those are the available online methods. A few are available, but offline are the most used. Although there are difficulties with offline, such as the one that we talked about just now. Let us look at offline measurements. Let us look at measuring the total cell concentration. Let us take one at a time. Yeah, I think that is okay. The total cell concentration, if you are considering a bunch of cells, consists of both live cells and dead cells. If we look at live cells alone, because the viable cells are the ones that are contributing to product formation and so on, we would want to follow the viable cell concentration. Sometimes the viability percentages are very high, 97, 98%, that it doesn't really make a difference whether you follow viable cell concentration or total cell concentration. But there is a difference in principle, and it can it can turn out to be important. The difference can turn out to be important. So you need to know that there is a total cell concentration measurement and a viable cell concentration measurement. Let us first look at the methods to measure total cell concentration. The first one is what is called OD, or optical density. OD stands for optical density. It's a historic term. It doesn't represent what it measures, but we will talk about it. You could also measure dry mass. Take sample, a large enough sample, dry it out completely, and then measure the mass. That is the mass, and that mass came from a certain volume. Therefore, mass of the cells by the volume gives you the total cell concentration. You could also use something called packed cell volume. This is the volume that is occupied by the cells in a rather concentrated solution of cells, typically in the industry. Okay. So this is the packed cell volume is typically used in some industries as a measure of the total cell concentration, the fraction of volume that is occupied by the cells. Or you could also use cell contents measurements, especially for molds. We said, if you remember, we said the molds were organisms that grow by extension of hyphae. So the number of cells may not be an indicator, it's just one cell, but increasing in mass through extension of hyphae. For such kind of organisms, we measure the contents of the cell. So those are the methods for total cell concentration measurement. Let us see the principles one by one. The optical density, the so-called optical density, is measured by using a spectrophotometer, but it is actually a measure of cell scatter. It is not a measure of absorbance by the cell. This is something that you need to remember very clearly. We have a cuvette here that is placed. Let us say that the cuvette has a cell suspension here filled till the measurement level. Then there is a certain wavelength that is shown on the cuvette. The cells, they are micron sized, so the Mie scattering range comes into effect. They scatter cells, and whatever light is not scattered is sent to the detector. That is measured. So that is measured and related to the incoming light to get the extent of light that has been scattered. The light that has been scattered is proportional to the concentration of cells. Okay. Make this difference very clearly set in your minds. This is cell scatter and not cell absorbance. Okay. Although the same instrument is used and something called optical density, the term is used, which is a complete misnomer. But anyway, in terms of the principle, it's a misnomer. It's a well accepted term. But keep this in mind. It is a measurement of scattering by the cells, and the cell scatters, the amount scattered is proportional to the cell concentration. That's the reason why we use about 600 nanometers. The principle is scatter. The scattering occurs maximally when the wavelengths are not absorbed by the cell. So we would like to work with wavelengths that are not absorbed by the cell. So anything in the range of 500 to 600 is actually fine. In fact, I have even used 450 in certain cases, so that's perfectly fine because you are measuring the extent of light scattered. 600 is a very typical number. You could use that. Anything between 500 and 600 is perfectly fine. Then what is done is using a calibration curve between the dry cell concentration. Remember, we did talk about dry cell concentration measurement. You take a large enough sample, you dry down the cells completely using a vacuum oven overnight, and then you measure the mass of cells that was present in that particular volume. Therefore, you have a dry cell concentration. Before you're going to dry that, you take a sample and measure the OD, and thereby you can relate dry cell concentrations, various values of them, to the OD, and you would get a straight line in a certain range, and that is the calibration curve. You could measure OD and get the relevant dry cell concentration using this calibration curve. That is what is typically done. The linear regime is important while measuring. You need to pay attention to that. The linearity, there are two aspects there. If you plot dry cell concentration versus OD, it is going to be linear till a certain point, and above a certain dry cell concentration, the linearity is going to break down. That's a law of nature. So it is going to be linear here, and above a certain dry cell concentration, it is actually going to saturate. We should not operate in this region. Of course, we measure, but we limit our range of operation to this linear region. In other words, we need to measure only in this linear region. If it happens to be in a higher OD region, we should actually dilute the sample to get the OD reading in this range, and then get the dry cell concentration corresponding to the measured OD and multiply it by the dilution factor to get the actual OD. If you follow all this, it's fine. Otherwise, just keep this in mind whenever there is a need for measurement. You go through this in detail or look at some reference book. You can always ask me to. There is another reason for the loss of linearity. That is because of the detector range. If the amount of light reaching the detector goes below a certain level, or if the amount of light that is scattered is very high, then the detector saturation happens. Then also there is a problem. So we don't work in the region of detector saturation. Depending on the spectrophotometer, I would feel very comfortable working only with OD values, say between 0.1 and about 1 or so. That way I am pretty much in the linear regime and in the normal detector region. Of course, this depends on the spectrometer, the maximum value, and so on and so forth. You need to worry about, during this measurement, interference by salts and substrates also, because we are taking a sample here, drying out the whole thing. When we dry out, whatever is soluble in the broth is going to contribute to the mass measurement. So the mass would contain, the mass that is measured would contain dry cells plus the salts plus unutilized substrate plus the other things. So we need to have an appropriate control by which we can account for the mass of salts and substrates and so on. So that we need to take into account while measuring OD. This is a finer point. I just thought I'll mention this here during the measurement. There is a video here. Let me show you the link, which can give you some more details. You may want to take a look at it. It's what is given in number 14, OD measurement. You can go and take a look at this video. Okay, let's move forward. The packed cell volume, the percent of total, this we mentioned earlier. If this is going to be your cell suspension, you spin it down using a centrifuge, and the cells are going to occupy this volume here. The total volume is this. So this volume by the total volume is going to give you the percentage packed cell volume. This is used in the industry. The next is cell contents measurement, especially for mold, which grows by extension of hyphae, not by division of cells. The amount of DNA, the amount of total nitrogen, the amount of protein, the amount of amino acid in cells could be measured. And if we assume that the percentage DNA per cell does not vary too much, then this becomes a direct measure of the cell concentration itself. Okay. This is the basis for the measurement of cell concentration through the measurement of its contents. The typical contents that are measured are DNA, total nitrogen, protein, and amino acids in that order of importance. DNA is usually preferred because it doesn't vary too much during the various parts of cell cycle and so on, before it splits up, of course. Total nitrogen could be measured, protein could also be measured, but there would be variations. Amino acids, there are some variations, but these are all accepted methods for cell contents measurement. Those were the methods for measuring total cell concentration. Now let us look at the methods for measuring the concentration of cells that are live, that are viable. Because the viable cells are the ones that are going to actually contribute to the functioning of the bioreactor. The methods are dye exclusion, plating, and others. We'll just look at these two methods. There are videos here. Before I get into the videos, let me just tell you the principle of dye exclusion and plating. Dye exclusion means or is based on the principle that certain dyes which can be visualized are excluded by living cells. So in the presence of dye, the cell is going to appear undyed, uncolored. So by counting the number of cells that are not colored and by counting the total number of cells, you can have percentage viability or the viable cell concentration itself from direct measurements. There are slides, specialized slides that can be used for these measurements. They are called Neubar chambers, hemocytometers as they are called, and there are ways by which you could reliably measure the number of cells in a particular volume and therefore the cell concentration. That is dye exclusion method principle. The plating principle is that you have an agar plate, a solid medium agar which contains agar, and then you take a certain low concentration of cells and plate them here, and assuming when you plate them here, the cells are going to multiply, and when they multiply to a certain extent, the colonies that arise from each cell become visible. One cell becomes 2 cells, become 4 cells, 8 cells, and so on. And as long as there are, let's say, millions of cells, that would form a colony. Okay, and at that stage, the colonies become visible to the naked eye, and you can count the number of colonies. Assuming that each colony arises from a single cell, you could count the number of colonies and relate them to the number of cells that were there in at the time when the plating was done. Okay. These methods take time. The plating method takes time. You'll have to keep it overnight and so on, even for bacteria which grow fast. But that is a method that can be used because colonies will arise only from live cells. So as long as you make sure that the cell concentration is measurable by counting the number of colonies, you could use this. The videos here, let me show you the details of the videos. You may want to look at them. For the trypan, the number 15 for the trypan blue exclusion method and 16 for plating for viable cell concentration measurement. These are nice videos that you can look at for the details of the measurement. For the substrate and product concentrations, spectroscopy, many kinds of spectroscopies can be used. Absorbance spectroscopy, which we normally know, the absorbance at a particular wavelength, which is particular for that molecule, is used to measure the concentration. Fluorescence we looked at, NADH fluorescence and so on. There could be fluorescent substrates, fluorescent products that can utilize this method. IR spectroscopy, infrared spectroscopy, we did talk of something related to that near IR. NMR, nuclear magnetic resonance spectroscopy, EPR, electron paramagnetic resonance spectroscopy can all be used. Spectroscopic methods can be used for offline measurement of concentrations of substrates and products. Chromatographic methods can be used. Gas chromatography, high pressure liquid chromatography, HPLC, and so on, they can be used. And there are many such methods that can be effectively used. I think we have come to the end of module 2. Very briefly summarizing what we did in module 2, we looked at biomass and bioproducts as two important outcomes of a bioprocess. That was the main theme of module 2. We looked at the details of these products and then the models for the rate of cell formation, simple models as well as some detailed models, and then what are substrates and how they are converted and how the substrate affects the growth rate. We had seen that, some models for that, and also when we looked at that, we showed that the monad model gives us the effect of substrate on growth rate. That's the simplest representation. There are other representations that are given here. And then for the product formation, we have the Ludeking-Piret model, Rp equals alpha Rx plus beta x, the growth dependent parameter, the growth independent parameter, and so on. Then we talked about a few concepts such as yield coefficients and maintenance. Then we looked at enzymes, some enzymes that are used in the industry, enzymes that can be used for bioconversions in a bioreactor. And we said that we need to know the kinetics of the enzymes. We looked at Michaelis-Menten kinetics in detail. We went through the derivation. Then we also looked at inhibition kinetics, the competitive, non-competitive, and the uncompetitive inhibition kinetics. We worked out a couple of problems based on those. And in this lecture, we looked at methods to measure the concentrations of biomass or cells, substrates, and products. We said that there were two different kinds, the online and the offline methods. Online preferred, but the reliability of the methods available are to a certain extent. Very few methods are highly reliable. Then we looked at the principles. We looked at methods for total cell concentration measurement, such as OD measurement, the packed cell volume measurement, the cell contents measurement method, and methods for viable cell concentration such as dye exclusion and plating. Finally, some methods for substrate and product concentrations we also saw. Okay. This concludes module 2, and this is lecture 9. When we meet in lecture 10, we will take things forward with module number 3. See you.